Okay, now what we are talking about DNA replication, correct? And in DNA replication, we are talking about how the replication process take place. So or both the DNA strand, they are complementary to each other, correct? So this is 5 to 3 direct, this is 5 and 3 end, yes. And the next
Yeah.
complementary strand, it will be here, it will be 3, and here it
Mm-hmm.
will be 5. Agree?
Yeah. Yeah.
They are the complementary strands. Again, remember. So once what we are saying, once the replication process has to take place, replication process is assisted by one enzyme, which is known as DNA polymerase.
Yeah.
Correct? That DNA
Mm-hmm.
polymerase, which is there, it will be attached on which side of the strands it will be attaching? 5 and 3.
Pardon? 5 prime to 3 prime?
I'm sorry?
5 prime to 3 prime direction?
Yes, 5 to 3 direction. Correct?
Yeah.
5
Yeah.
to 3 direction. So actually what happened, DNA helicase will be there and it will be opening up the DNA from here. The 2 strands of DNA get open up from here. Oh, sorry,
Yeah.
it will get open up from here only. Okay. So once it will
Mm-hmm.
get open up from there, just see how it will look like. Now it will be like this. Okay.
Mm-hmm.
Like this. And the other strand, because one, it will be like that. Okay. So if this was 5, so
Mm-hmm.
this will be 3 and this will be 3. So down it will be 5.
Yeah.
Just hold on. This is 5. Now, DNA polymerase, it will get attached to the 5th end, yes? Where
Yeah.
is the 5th end here? So obviously it will start to replicate from here, like
Yeah.
this. Yes? So it will
Mm-hmm.
start to make a DNA strand like this, correct?
Yeah.
The other, this is 3 direction. What will happen
Mm-hmm.
here?
It will
It will
like
be
go in
moving from
fragments.
down, from, we call
Up.
it replication fork. Please make note of this.
Yep.
This is called replication fork.
Mm-hmm.
So this first strand, we call this strand which is moving towards the replicating fork, that is called leading strand. Please make note of this.
Yep.
Leading strand. So what is a leading strand? A leading strand is a new strand which is moving towards the replication fork. Okay.
Yeah.
Now the
Mm-hmm.
other strand, which is the other way around, because it cannot start from here. It cannot start from here, isn't it? So it has to,
Yeah.
so it will start from here, wherever the 5 will be there. 5, it will
Mm-hmm.
start from here, and it will move away from the replication fork. This is
Yeah.
the most important thing. So that strand will be called what?
The lagging strand.
Lagging. Shabash. Lagging strand. So in this way, once one by one, this replication fork or this area, it will keep on opening. The leading strand, it will continue to move in a straight line like this. Yes, it will
Mm-hmm.
continue to move in the straight line. Ultimately, the full leading strand will be replicated. Correct? While
Yeah.
the lagging strand, what will happen to the lagging strand? Lagging strand will be making different components, different
Yeah.
strands. For example, for in this case, how many strands will be there? There will be
3.
4.
Oh, 4. Yeah.
There will be 4 strands. So there will be
Mm-hmm.
4 lagging strands. Okay.
Yeah.
These
Mm-hmm.
4 lagging strands, they are also known as
Okazaki
Okazaki.
fragment.
Okazaki, very good. Okazaki fragments. So these Okazaki fragments, once they will be, the replication process will get over, the primitive, the old DNA strand, they will get entangled again. Yes.
Mm-hmm.
And the new strands will be made. I will be talking about a replication process, where what is conservative and semi-conservative. Yes. So anyway,
Okay.
the point what is happening here, these Okazaki fragments later on they will be joined together by DNA ligase.
Yeah.
Correct? Okay. So
Mm-hmm.
in this way, the replication process tend to take place. So I think everything is clear. Yes.
Yeah.
Now, once we are talking about replication process itself, now there is what kind of replication process is there, whether it is conservative, semi-conservative, or dispersive. Okay, how the
Mm-hmm.
replication process take place. You understood everything and you wrote it on your notebook. Yes.
Yes.
Okay, I'm just leaving it from here. Now what I'll do, just hold on. I'll stop recording. Okay.
Okay.
Now, next thing is, once we are talking about conservative and DNA replication process, okay?
Mm-hmm.
Okay, once we are talking about DNA replication, now in the DNA replication process, there are three theories, okay? How the DNA
Mm-hmm.
replication takes place. One is called conservative model of replication, semi-conservative, and
Mm-hmm.
dispersive. Do you have any idea if you can share?
What?
Do you have any idea what you can share with me? Do you have any clue about this?
Yeah, isn't DNA replication semi-conservative?
What does it mean?
It means that one strand of DNA is coming from the original strand of DNA and then one strand is new.
Very nice, very nice. So this will be conservative,
Mm-hmm.
semi-conservative. How you can prove it?
Pardon?
How we can prove it that DNA replication process is conservative, semi-conservative, or disruptive?
I mean, conservative means that both strands are the same as the parent strand, or like the two
Exactly.
strands are the same.
Uh,
So, but I
yeah.
mean, that doesn't really happen because like one strand is new.
So actually, for example, these are the two strands of the parental strands. So they will make completely new strands of DNA. Yes.
Yeah.
And the
Mm-hmm.
original remains as such. And it is
Yeah.
not possible. Correct?
Yeah. Mm-hmm.
Okay. Now, next thing is what? Next thing is
Semi-conservative.
semi-conservative. Semi-conservative, if we take the same model, it will be like this.
Mm-hmm.
Okay, the original one.
Mm-hmm.
So once it will replicate, it will generate in this form. Yes. One will be
Yeah.
this.
Mm-hmm.
Okay. And one more will be like this.
Mm-hmm.
Correct? Is it true or false? I think it is the way only.
Yeah, and then
Here
the
actually, here there will be one more also. Yes.
in conservative, wouldn't they both be the same as the parent strand of DNA?
No, no, wait. I will tell you. I will tell you. What is the confusion here? No, it will be like this. Yes.
Yeah.
So here, actually, here it should be the second one also like this.
Mm-hmm.
Oh, sorry. The second one will be like this,
Pink.
isn't it? So in this way,
Mm-hmm.
this was conservative. This is semi-conservative. What about the disruptive?
That would be that like it's
Some
all
part will be
random. Yeah,
within one
some part would
strand.
be, yeah. Some parts
Exactly.
would be old, some parts would be new.
Exactly. So it will be like this. So we can
Yeah.
continue like this. Yes.
Mm-hmm.
So it will be dispersive.
Yeah.
Okay. Now, what is the experiment which will be proving that DNA replication process is semi-conservative? This is a typical question in exam. Okay. Now for that, do you have any idea about this?
I don't know what the experiment is.
Okay, there is one a Messelson stall experiment is there. Okay.
Oh, it's stall?
Yeah.
Yeah, I think I know it then.
Then look here. For example, nitrogenous base that we are taking N14. Yes.
Mm-hmm.
So for example, two strands of DNA are there, and these two strands of DNA are there. Correct?
Mm-hmm. Yeah.
After these two strands of DNA, they have N14. They are made up of N14. We put
Mm-hmm.
into these strands of DNA into a medium which contain N15.
Yeah.
So once N15 will be there, then what will happen? It will make a strand. For example, a new strand is made like this, okay?
Mm-hmm.
Semi-conservatively. So one strand which is original, it will be having 14. And the other strand, which is the other one, it will be having 15 molecular mass, correct?
Yeah.
So once we use the centrifuge,
Mm-hmm.
that a settlement in the centrifuges, yes, then what will
Yeah.
happen? It will give us a rate in between N14 and N15.
Mm-hmm.
It will be having, it will come here this way.
Yeah.
So it proves that DNA replication process is semi-conservative instead of conservative. If it is conservative, if N15 is there and new strand which are made, the baby strand, they should fall into this range, or daughter strand.
Yeah.
So, but actually they are not falling here, nor here, and they are falling in between. What does it mean? One strand is 14 and the other strand is 15. So it is coming in between 14.5.
Yeah.
Okay.
Mm-hmm.
Now, this is the first thing. Now, replication of DNA, it is revised, okay, completely. Now, next step goes, next step is what? The process of transcription and translation, correct? Now,
Yeah.
in the process of transcription, so the protein synthesis is a process which is divided in two stages, transcription and translation, yes? So what is there?
Yes.
Like DNA, it first, it is
Mm-hmm.
during transcription, what happened? DNA get transferred into RNA.
RNA.
Which RNA precisely? Messenger
mRNA.
mRNA. And this RNA get transferred into,
Amino
translated
acids.
into protein,
Protein.
amino acid. Yes.
Yeah.
This is known as central dogma.
Mm-hmm.
What does it mean then? Central dogma means this is the reality. This is the process where DNA will get converted into RNA and RNA will get converted into protein. This is the basic structure of protein synthesis. The core of the protein synthesis. So now once we are going into this detail, before going into the detail of transcription and translation, RNA, once we are talking about the RNA, there are how many types of RNA? There are three. Like you said, mRNA, messenger RNA. What is the role of this messenger RNA?
It's to bring the RNA to the ribosomes.
No. Role is it takes the message from DNA to the RNA that transcribe me or translate me. So it is a messenger from DNA to ribosomes to make protein code.
Yeah.
Correct? Second part is, what is the second part? rRNA.
Yeah.
What is this?
Yeah.
What is the
It's,
main part of this thing? Ribosomal
it's
RNA. What is the function
yeah.
of this ribosomal RNA?
It reads the mRNA so it can translate it into the amino acid sequence.
No. rRNA is actually structural unit. It combines with proteins, okay, to make ribosome. So that is why we call them ribosomal RNA. Again,
Okay.
very important. Ribosomal RNA, it combines, look here, for example, if you, ribosomal RNA is what? It is made up of RNA, obviously, plus proteins.
Mhm.
Once they combine together, they make ribosomes.
Yeah.
Okay. So ribosomal RNA is actually structural part of proteins.
Yeah, so it
No,
reads the
it make the structure of ribosome.
yeah, so
Structure of
it reads
ribosome.
the mRNA and translates those proteins, those amino acids to make the ribosomes.
Come again.
Yeah, it reads the mRNA and translates that into the amino acids, which then makes the ribosomes. Right?
No. It's a confusion then. Look here, reading the mRNA. This is the duty of ribosomes.
Mm-hmm.
This is, please make note of this. Reading the this one, what to say, the messenger RNA, this is the function of ribosome. Ribosomes are responsible to decode messenger RNA.
Mm-hmm.
Okay. rRNA has nothing to do with this thing.
Okay.
rRNA is what? rRNA is actually why we are calling it ribosomal RNA, because this is the part structure of ribosome.
Okay.
So ribosomal RNA and proteins combine together to make ribosomes.
Mm-hmm.
So there, now you get my point. So
Mm-hmm.
don't mix up with this thing like messenger RNA will be read by ribosomal RNA. No. Reading the messenger RNA, decoding the information from messenger RNA, this is the duty of ribosomes. Now you can
Mm-hmm.
say, because since rRNA is the integral part of ribosome, so in one way I'm correct, because rRNA is present inside the ribosome. Yes, because ribosomes are
Mm-hmm.
made up of rRNA, correct? But please
Mm-hmm.
remember, in a different way the statement gets true, okay? But
Mm-hmm.
please remember the major function of ribosome to decode the information of messenger RNA. This is the responsibility of ribosomes.
Mm-hmm.
Okay. Now, rRNA we discussed now. Third part is transfer RNA. Yes, tRNA.
Mm-hmm.
Now, what is the role of tRNA?
It brings the amino acids to the ribosomes so it can create the
So,
protein.
yeah, it brings the transfer RNA main responsibility is to transfer amino acid to the protein synthesis site. What is the
Mm-hmm.
protein synthesis site?
Ribosome?
Very good, ribosome. So it will bring, so once the ribosome, they are decoding the messenger RNA, transfer RNA has the responsibility to what? To transfer required amino acid which have anticodons, so they will transfer at the protein synthesis site. Now this is first thing that we are done. Okay.
Mm-hmm.
Now next thing, what will, this was the basic thing. Okay. One more thing, because this thing later on I don't need to teach it. Transfer RNA are highly specific for their amino acid. Each
Mm-hmm.
transfer RNA is fixed for one particular amino acid.
Yeah.
Correct?
Mm-hmm.
Each transfer RNA is fixed. For example, there are 20 essential or 22 essential amino acids. What does it mean by essential amino acids?
It means that our body can create them.
No. Essential amino acids are those amino acids which are
Oh,
basic.
that we can get from food. Okay.
No. Say
That are basic and are found in our body.
no. The essential amino acid, normally we call them, why we call them essential amino acid? Because they are part of all living organisms. Plants, animal, bacteria, fungi, everything, all the living organisms, they are the integral part of them. Okay. Apart from this, then other amino acid will be there. Like, so how then one living organism is different from the other? Because these essential amino acids, which are 22 or 20 20 amino acids, these amino acids, they join in different formations, arrangements are different, so they give different structure. Like plants have different kind of structure, animals have different kind of structure. Why? Is amino acid are the same, but their arrangement is different, so that is why they look different. It is just like an example of houses. Brick is the same, but arrangement of the brick and architectural structure, it make the two buildings which are made of the same material different. Correct? Same is the case for essential amino acids also. I hope you got it. Yes.
Yeah.
I'm moving next now. Next thing is, once we are talking about protein synthesis, in protein synthesis, first part is transcription. Yes. What is transcription?
Transcription is where mRNA is formed from like the template strand in DNA.
Okay, now the point is good answer. Okay, but the point, what I normally, once I'm teaching to any kid, okay, I give making messenger RNA from DNA is called transcription. If you will write this statement into your exam, inshallah, you will not get any mark. But this is just for your memory. What is that thing? Formation of messenger RNA from DNA. This is called transcription. Okay, this is the basic definition. Okay, you cannot get marks for this. This is for our understanding. So making messenger RNA from DNA, this is transcription. But if we put the things into correct words, okay, biological term, so the process, transcription, the process in which the genetic information from the DNA is transcribed in the form of messenger RNA inside the nucleus.
Yeah.
Can you repeat?
Pardon?
Can you repeat?
The process of forming
the process in which genetic
messenger
information
RNA
from DNA is transcribed in the form of messenger RNA inside the nucleus. Once you are done, please tell me.
Okay.
Now, transcription. So from this definition itself, we come to know that transcription is the process which takes place inside the nucleus. So two processes, transcription takes place inside the nucleus, while the translation takes place out of the nucleus in cytoplasm. Correct? Okay.
Yeah.
During the process of transcription, now what happens? Like already you took the name. One is like this, yes? Two strands of DNA are there. So once they will get with the DNA helicase, they open up. One strand.
Doesn't DNA polymerase do doesn't RNA polymerase do everything in transcription?
Polymerase is responsible to make strand, but opening opening up the this helix need DNA helicase.
Oh, but what we were taught in school was that RNA polymerase acts as all of the enzymes that are needed in DNA replication. So RNA polymerase acts as DNA helicase, where it opens up the strands and then
RNA polymerase act as a DNA helicase.
Yeah, that's what we were taught.
Oh my goodness. This is a new kind of, thoughts that you are
Or
giving.
maybe I heard it wrong, but okay.
No, you confirm it. It is not correct. How
Okay.
the DNA, look here. Have you ever heard of a term which is called transcriptional bubble?
Yeah.
What is
No, wait, no. No, not
For example,
transcriptional
now
bubble.
look here.
I've heard
No,
of
I
replication
will tell you.
bubble.
I'll tell you. Look. For example, this is the DNA, okay? The whole DNA.
Mhm.
And from this
Yeah.
DNA, this part of the DNA is responsible for one particular enzyme. So
Mhm.
you don't need to open up from here, or from here, or here. What will
Okay.
happen? The DNA helicase will open from here only. Once these two
Mhm.
strands, they will open up, they'll get unzipped. So it will take a shape like this.
Mhm.
It will take a shape like this. Okay.
Mhm.
So this is called
Yeah.
DNA transcriptional bubble.
Mhm.
Okay. With this transcriptional bubble, why? Because this was the genetic information which we needed. We need this enzyme. This enzyme is present in this area only. We don't need to open up the other DNA.
Mhm.
Correct? So in order
Yeah.
to open DNA, unzipping DNA, you need helicase. Now,
Yeah.
this is a new information for me, like what you are saying, that RNA
Mhm.
polymerase act as a DNA helicase. I have never heard of this.
Okay.
Yeah, so I will reconfirm and then I'll get back to you also. But please try to reconfirm from your end also. Okay. To
Okay. One minute.
to uncoil, the property of DNA polymerase and RNA polymerase is the same. Yes or no?
Pardon?
The quality, the properties of
Mhm.
DNA polymerase and RNA polymerase is the same. Both
Yeah.
are responsible to replicate. So if the RNA polymerase has the ability to act as a DNA helicase, so DNA polymerase must have this ability. Why we are using in DNA replication, DNA helicase, but here once you are unzipping the DNA, you are not required. Very important question. Yes or no? It makes sense.
Mhm.
Just check. Okay. Next thing is
Okay.
what I was saying. If we say these two strands of DNA,
Pardon?
if we have these two strands of DNA, yes,
Yeah.
one strand
Mhm.
act as a coding strand and other strand act as a template strand.
Yeah.
Coding strand is also known as sense strand. Please write down.
Mhm.
Coding strand is also known as sense strand. And template strand is known as anti-sense strand. Okay,
Uh-huh.
anti-sense.
Yeah. Yeah, I think, sorry, going back to our discussion, I think RNA polymerase unwinds and rewinds the DNA. So in transcription, DNA helicase is, or any helicase is not required.
So that is why, you know what I mean, that is what I told you. Now you are referring to some stuff from yourself. What you
Yeah,
are saying,
I
you are,
looked at my notes, like the notes that I took, and it said that are in transcription, helicase is not required. And then I quickly looked online and it said, yeah, RNA polymerase acts as helicase as it can unwind and like rewind the DNA.
okay. So RNA polymerase in your, according to your notes, it is acting as helicase as well as gyrase.
Yeah, so in transcription, the only enzyme needed is RNA polymerase. It takes the role of all the other enzymes.
Finger crossed. I'll get back to you once, I will not confirm or deny now. Okay.
Okay.
There's a conflict. Yes. So I will
Mm-hmm.
give you the answer once I will confirm myself. Okay.
Okay.
So you stick to your point, whatever you have written in your
Okay.
notebook. You will stick to your notes now. Why? Because it is from your teacher, correct? Please
Yeah.
remember, Muhammad Anwar wording matters nowhere, okay? Once it comes to this thing, until and unless there is one proven kind of thing, okay? So whatever
Okay.
the teacher have given you, follow that thing, okay?
Okay.
Until and
Yeah.
unless I tell you, no, it is not correct and blah, 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 correct?
Okay.
So this is,
Yeah.
so anyway, this thing for the time being, we are settling it down, okay? So now
Okay.
we are getting back to the same thing. One is coding and template strand. Coding strand is also called sense strand, while template strand is also called as anti-sense
Anti-sense.
strand, correct? Okay. Now, the point is why we are calling coding strand as sense and why we are calling template strand as anti-sense strand. Any explanation?
I'm not sure.
Okay, the point is coding strand can use it. You know what is happening actually? During the process of transcription, the coding strand reads the information from template strand and directs RNA polymerase to make a sequence of nucleotides. So so what exactly is the transcription? Transcription is the mirror image of coding strand. Please, this word you have to remember. Transcription is the mirror image of coding strand.
Okay.
Okay, once we are talking about why it is a mirror image, I will tell you. Now we have two strands now here. Yes, strand A and strand B. For example, this is strand A and strand B. Which
Yeah.
strand will be acting as, and why we are calling it coding strand or sense strand? Because coding strand is using its senses, because it reads the information and guide the this RNA polymerase to make sequence of nucleotides, correct? And those
Yeah.
nucleotide sequences are complementary to what?
Which,
RNA polymerase will make the
yeah.
sequence of nucleotides, yes, of messenger RNA. And that messenger RNA nucleotide, they will be complementary to what?
The template?
Very good, very good. Whether it was a fluke or it was well thought answer.
No, I know.
Okay, good. So it will be the complementary, it will be complementary strand to the template strand. Yes, it will be complementary to the template strand. Now we have two strands of DNA here. Yes, this is A and this B. Which strand is acting as a template strand and why?
Wait, I'm confused. Is the template strand the strand that is being coded from, or is the template strand the strand that is needed, like the information on that strand is needed?
Now I feel there will be again conflict, you know, for
No,
me.
it's just for clarity.
No, it is not. I'm saying it. Template strand, it is acting as a template. Yes, a template strand is a strand on which the messenger RNA will be combined together.
Okay.
You understand? So now just give the idea like whether A is A is the template or B is the template. And if you are giving this as A and B as a template strand or B, reason.
Would B be the template strand?
Okay, B be the reason.
No, wait. Okay, so the template, no, because the wording is a little different. Like, I forget what, like, for the coding strand,
Yeah.
like, is the coding strand the strand that we need the information from?
For example, I will tell you, I will give you the easy thing. Okay, just 1 minute. For example, if it combines here, this messenger RNA. So this will be acting as template strand. Template strand is a,
Okay,
you remember template,
so template
messenger
strand, so we,
RNA has to attach with template. You get my
yeah,
point?
because we need, yeah, okay, I get it now, yeah.
Okay, good. Now tell me, A is template or B is template?
B would be template because we need the basis from A.
Okay, B will be the template strand and A
Yeah,
will be coding strand, correct?
yeah.
Can you make a messenger RNA?
What do you mean make
Can
a messenger?
you make a messenger, draw the messenger RNA on my screen? You
Okay,
can. No, actually, how much time is remaining? It is more less than
Less-
1 minute, yes.
yeah,
Wow. Okay. Yeah. Just try to do it fast because then one more thing. There will be a session break for 5 minutes
okay,
and then again it will restart. Okay. Because I cannot immediately start the meeting. Now I think some changes are there in Zoom. Okay. So there'll be
okay,
4 minute, 5 minute break is there. Then I can restart the meeting. Okay. So bear
okay.
with me. Yeah. Tell me. So you have
Is,
started from here and it is coming down. Yes.
yeah,
Okay. To be continued because a session is going to end.
yeah.
Okay. So I'll
Mhm.
see you